Our next gram-positive rods belong to the genus Listeria, and the species that we'll talk about is Listeria monocytogenes. Listeria are non-spore-forming aerobic bacterium. They are uniform in shape, so they will always look like a small rod-shaped bacterium. Under a microscope, that helps to distinguish them from another genus that we'll talk about later called Corynebacterium. Corynebacterium can be what we call pleomorphic. Sometimes you can see different shapes even though it's the same species. They are short rods, which can distinguish them from bacillus, tend to be a little bit longer rods. What makes Listeria an effective pathogen in humans is the fact that it can grow at a pretty wide temperature range. It can grow anywhere from 1 to 45 degrees Celsius, meaning that it can not only survive being refrigerated, it can grow and replicate in the refrigerator. It has beta hemolytic activity, and we generally see Listeria infections as foodborne illness. While it may not be as well known as things like Salmonella or E. coli, Listeria monocytogenes is actually the third most common cause of fatal foodborne infections. So it may not be as common, but it is very dangerous to get as a foodborne illness. Listeria monocytogenes has a really interesting lifestyle. It will attach to the host cells, so perhaps you ate it, whatever, it got into your body. It will attach to your cells and allow itself to be phagocytized. It produces two enzymes, listeriolysin O and phospholipase C. Those allow it to escape from the phagolysosome. So it doesn't want to be in there and be digested. It allows itself to be phagocytized, but it will then escape and just be in the cytosol of the cell. In the cytosol, it can replicate, and then from there, it has to move from cell to cell to cell. Now, if you're a bacterium and you need to go from one cell to another, there are a couple ways you could do it. The more dangerous way to do it is to go out of the cell you are currently in and go over to another cell. And the reason that that's dangerous for you as a bacterium is because it exposes you to the immune system. And so what Listeria does instead as a type of immune evasion mechanism is it uses a protein called ACT-A, and that protein rearranges the actin in the host cell membrane. This allows Listeria to literally push itself into the neighboring cell so that it can move from cell to cell without risk of detection by the immune system. They have a gene called positive regulatory factor A, PRFA, and that controls the expression of the genes necessary for escape from the phagolysosome and for migration from cell to cell. Here you can see this poor guy is eating some food that is going to lead to a listerial infection. The cell binds to your host cell, it gets internalized, it makes listeriolysin O, phospholipase C, it escapes from the phagolysosome, it upregulates a couple of other proteins including ACT-A, that allows it to push itself into the next cell where it almost forms its own little phagosome, which again it can escape from to replicate. So it's a pretty interesting life strategy, and it allows Listeria to be a very effective pathogen once it gets inside the host. The bacterium can be isolated from environmental and fecal sources. Generally, infection comes from consumption of contaminated food. We are most often talking about animal or dairy products, but Listeria has also been associated with produce. An infected mother can also pass the infection, or a pregnant mother, I guess, can pass the infection to the fetus. As I mentioned earlier, this is not a common foodborne infection, but it does have a high mortality rate. So here we're showing some of the diseases that can manifest with a listerial infection. So in neonates, a pregnant woman can have a spontaneous abortion if the fetus is infected. The child, the infant, could have abscesses and granulomas, meningitis, septicemia. In an otherwise healthy adult, you may experience flu-like symptoms, chills, fever, some nausea. In pregnant women and other patients with immune deficiencies, you can experience bacteremia or disseminated disease that can cause meningitis. And so the reason pregnant women are kind of grouped in there with people who have immune defects is because pregnancy does suppress the immune system. Pregnancy is a condition that makes individuals much more susceptible to infection. It's very hard on the body because a lot of the resources are devoted to growing an entirely new human being. It's a lot of work for the body to do, and so when the body is focused on doing that, growing the person, it focuses less on things like maintaining the immune system. And you'll notice here it specifies patients with cell-mediated immune defects. So there are two types of adaptive immunity. You have humoral immunity, and this is your antibodies, basically. And you have cell-mediated immunity, which is going to be your T cells. So humoral immunity, or your antibodies, are only going to recognize Listeria if Listeria is floating around accessible to antibodies. Antibodies don't normally go into a cell looking for an antigen. So it really doesn't matter if you have plenty of antibodies or zero antibodies. Listeria is hiding and not going to come into contact with the antibodies. But your T cells, your cell-mediated immune system, is critically important for helping you to clear intracellular infections. Your T cells are the ones that will know that you have an intracellular infection. So in a healthy individual who has functioning T cells, those T cells will be able to find the infected cells and tell them to undergo apoptosis, right? So the cell will be destroyed. Patients who don't have functional T cell immunity cannot fight off intracellular infections. So people with a cell-mediated immune deficiency, a lack of T cells or decrease in T cells, are going to be at much higher risk for all sorts of intracellular infections, including Listeria monocytogenes. So that's why we always recommend that you take a class like immunology either before this class or in tandem with this class so that you understand why I say these things. It's not essential. I've had plenty of people do very well in this class without immunology, but it is very helpful, and it puts a lot of this information in context. Listeria is not super fastidious. It will grow on most kinds of media, but one way, if you suspect a foodborne illness, to isolate Listeria as opposed to any of the other agents that can cause foodborne illness is to use cold enrichment. Again, this bacterium will not only survive being refrigerated, it will grow and replicate. So by putting your plate at a cold temperature, you would inhibit the growth of E. coli or Salmonella, and you would select for the growth of Listeria if Listeria were present in your original sample. Listeria is also motile, which is different from other gram-positive rods. So there are ways to differentiate Listeria from some of the other gram-positive rods. In cases of suspected outbreaks, scientists often use whole genome sequencing to track the source of the infection. It can be treated with gentamicin and penicillin or ampicillin. One of the things you have to consider when you're prescribing antibiotics for a Listeria infection is that the antibiotics have to be able to get into the eukaryotic host cell. If you have an intracellular infection, the only way to get rid of it is to get the antibiotics into the cell where the bacteria is. So things like gentamicin can get into the eukaryotic cell. So that's something that as a physician, you would have to consider. You should avoid raw or undercooked dairy and other animal foods. Really, really common in dairy, cheeses, yogurts. In fact, the summer that I moved to California, when I got this position, the summer of 2015, I was still living in San Antonio, finishing up my postdoc, and there was an outbreak of Listeria monocytogenes associated with ice cream. The specific brand of ice cream is called Bluebell ice cream. No lie, like the best ice cream ever. Bluebell ice cream is so good. But they pulled it all from the shelves because one of the processing plants, at least one of the processing plants, was contaminated with Listeria. And I was so bummed because it's really hot in Texas. Not as hot as it gets here, but it's humid, and ice cream is just so good. And if you're going to get ice cream and eat all those calories, it might as well be good ice cream. But then I found out that at least 3 people died of Listeria monocytogenes infection from eating ice cream. And so I was okay that they pulled it off the shelf. I missed it, but I didn't want to die. So, avoid raw or undercooked dairy. This is also why I went to Sprouts once and they had a sign about drink raw milk, it's so great for you. And I was horrified. And then they said, and you can give it to your babies. And I was even more horrified. Please do not give raw milk to your babies or your children. If you want to drink it, that's on you. I don't think you should, but you didn't ask me. It is not safe for infants to drink raw milk, so please don't do it. This is one of the reasons why. You should also thoroughly wash all produce. It's been a while, but it's probably been at least 10 years now. Maybe not quite that long, but I definitely lived in Texas at the time. There was also an outbreak of Listeria associated with, weirdly enough, cantaloupe. So think about it this way. Cantaloupe, delicious. Got that really heavy rind, right? You cut through the rind and you've got that beautiful, juicy, delicious fruit on the inside. How many of you, when you buy a cantaloupe, actually wash the rind of the cantaloupe? You're not going to eat the rind, so why do you wash it? You only eat the stuff on the inside. I mean, maybe if it had big visible chunks of dirt or something on it, you would wash it, but for the most part, most people, when they buy a cantaloupe, pick out a nice cantaloupe, test it, make sure it's ripe, take it home, put it in the fridge. Get your big old knife, cut into it, and store it in the refrigerator. And so what happens is animals defecate in produce fields, and you may not even see the feces, but they're there. There could be bacteria in those feces, and when you take that big old knife and you cut into that cantaloupe, you're pulling whatever's on the outside onto the inside. If it was something like E. coli, it probably wouldn't be that big of a deal because E. coli doesn't really grow in the refrigerator. But with Listeria, what you've basically done is taken this bacterium from the outside of the cantaloupe, brought it to the inside, the part you're going to eat, put it back in the refrigerator, and that bacteria is like, yes, this is my perfect temperature, and it grows and replicates, and then you eat that cantaloupe, and you can make yourself very sick. So thoroughly wash all of your produce, including your cantaloupes. Here are some other headlines. Protein bars recalled for Listeria risk. At sentencing, cantaloupe growers apologize for deadly Listeria outbreak. Texas-based Bluebell recalls 2 ice cream flavors over Listeria concerns. They eventually pulled all their ice cream off the shelf, not just 2 flavors. Okay, so maybe not an infection you've heard a lot about, but definitely an important one. The next bacterium we'll talk about is Corynebacterium diphtheriae. This is the causative agent of a disease called diphtheria. Diphtheria is caused by a gram-positive rod-shaped bacterium that grows in short chains, and so that's what they're showing you here in this image. They form rather large colonies on a plate, and the bacterium can be found worldwide. There are very low rates of infection, though, in countries that have high rates of vaccination. So Corynebacterium diphtheriae is a bacterium for which we have a vaccine. And I kind of hesitated there because we don't actually have a vaccine against the bacterium itself. I'll tell you what the vaccine is against in just a minute. So this was a headline that I saw probably 2 or 3 years ago, the first case of diphtheria in Spain since 1986 after the parents shunned vaccination. So the first case in over 30 years because the parents did not believe in vaccination. Their 8-year-old son got infected with Corynebacterium diphtheriae, developed the disease diphtheria, and unfortunately died. So this is a scary infection. However, it's relatively uncommon now, at least in parts of the world where we have vaccination. So I told you on the previous slide that we do have a vaccine, and then I said, well, sort of, it's not actually against the bacterium. The vaccine we have is against the toxin. So diphtheria is another one of those infections where the disease itself is mediated only by toxin production. So there is an exotoxin that some strains of diphtheria can produce. Those strains at some point had to be infected by a bacteriophage. Remember, a bacteriophage is a virus that infects bacteria. It can sometimes give them new genes, that process of transduction. So if the Corynebacterium diphtheriae has picked up the genes for toxin production, it generates an AB toxin. So B is the binding domain, A is the domain that gets internalized. So you see here, the binding domain binds to a particular receptor on the outside of the cell. That forms an endosome. The endosome is eventually acidified, which causes the toxin to dissociate from the receptor, and then it can exit the endosome. The binding domain falls off. The A, or active domain, its function is to interfere with protein synthesis. So specifically, it interferes with elongation factor 2, which prevents elongation of proteins. When a cell can no longer make its proteins, the cell dies. So the subunit inhibits protein synthesis by binding that elongation factor 2. Specifically, the binding domain binds to a heparin binding epidermal growth factor. And this is found on many, many eukaryotic cells, particularly on cardiac and nervous system cells. So diphtheria can become very, very dangerous if the toxin reaches the nervous system or the cardiovascular system. Generally, diphtheria is considered to be an upper respiratory infection, but again, if the toxin gets out and gets somewhere it's not supposed to be, that can be very dangerous for the patient. So generally a respiratory disease, what you will often see in the patient is this production of exudate. They're trying to show in 2 different patients, this production of exudate in the back of the throat. We sometimes call that a pseudomembrane. It almost looks like a little membrane, but if you tried to scrape this off underneath, it would be raw and bloody because what's happened here is tissue destruction because of the toxin. So after 2 to 4 days of exposure, a patient may develop symptoms that would include tiredness, sore throat, and the production of this exudate, and that forms that pseudomembrane. If the toxin is able to get into the bloodstream and get to the heart, it can cause inflammation in the heart because of destruction of cardiac cells, and also neurotoxicity because of destruction of cells of the central nervous system. So that pseudomembrane is really diagnostic. I would imagine that those of you who are going to practice medicine in the United States, I would put money on it that 95% of you will never see a case of diphtheria. But if you do, that single pseudomembrane is really, really diagnostic of the infection. In children, that pseudomembrane can be very dangerous because it actually can grow so large that it can cover the trachea and cause respiratory failure. So you have to be very, very careful in your patients with diphtheria. There is also a potential for a cutaneous form if the bacterium that produced the toxin get into a wound, but that is much less common. It can be diagnosed by culture, by nucleic acid, or by other biochemical assays. But again, for the most part, that pseudomembrane is unique. So even though diphtheria should not be a disease that you see, if you just remember that that pseudomembrane could potentially be diphtheria. A patient who has been known to be exposed or has developed symptoms of diphtheria can be treated with a diphtheria antitoxin. Generally, when we talk about antitoxins, and we will talk about them a few more times, especially in the bacterial section, antitoxins are preformed antibodies. These preformed antibodies are most often generated in an animal. And so what can happen is, because the toxin is the most dangerous part of this infection, if you give a patient antibodies which will bind and neutralize the toxin, you can help eliminate the development of further disease symptoms. So you're basically preventing the toxin from working anymore. So prior to a diphtheria vaccine, using diphtheria antitoxin, sometimes called serum, was the way to treat infection. You had to wait for the patient to develop symptoms, or you had to have a known exposure, I guess. Then you would give them the antitoxin to help neutralize and prevent the development of symptoms. The infection can be treated with standard antibiotics like penicillin or erythromycin. And now we have a vaccine. It's called the diphtheria toxoid. So it's a molecule that looks a lot like a toxin. It is not the toxin. And it tricks the immune system into making antibodies against the toxin. This is part of the Tdap or DTaP, depends on which one you get, vaccine. So that's a vaccine for diphtheria, tetanus, and pertussis. So we'll talk about those other two infections later. So we do have a vaccine. It is very, very effective, and that's why we have very low levels of diphtheria in countries that have high rates of vaccination. Some of you may have heard of the Iditarod. And if you already took microbiology with me, you've already seen this, but you're lucky and you get to see it again. The Iditarod is a dog sled race that occurs in Alaska, but it actually has some infectious disease links. And so what happened was, in 1925, there was an outbreak of diphtheria in Nome, Alaska, which is up here, like in very, very close to the Arctic Circle. And this was in January. So as you can imagine, all of this water up here was frozen. There were lots of big chunks of ice. Everything up here was frozen. There weren't really a ton of planes, and there weren't planes that could fly in that kind of weather. So all they really had was a railroad, which unfortunately you see here, the railroad went from Seward, Alaska, which is down here in the Gulf of Alaska, up to this town called Nenana, and then up to Fairbanks. So to get to Nome, though, there was just this one road, which of course was snowed over, so there was no way to drive or anything. And so when these children in Nome started showing up with diphtheria, they were requesting antitoxin, and there wasn't any. There was no serum. And so they called to Seattle, and Seattle had some serum, but they couldn't ship it up here because of the ice, right? And they couldn't fly it because there were no planes to land. So what they said they would do was go ahead and ship it to Seward, because the Gulf of Alaska doesn't freeze. But it takes a long time, and it took even longer in 1925 for a boat to get to Seward. they ended up finding another stock of some serum, I believe in Anchorage. So they sent it by train up to this Nenana, and then they did a dog sled relay to get the serum all the way to Nome, Alaska. And they were able to do that, and there was very little impact of diphtheria in the population because of the access to that serum. So it was very lucky that they were able to get that serum there. So the Iditarod now starts down here, but it does travel much of the same route. So it's kind of that celebration of being able to save the children of Nome, Alaska. You may have heard of the dog Balto. He's real famous, but he actually didn't run the most of the race. Another dog called Togo ran a much larger portion of this trail. So if you ever go to Alaska, I highly recommend it. You totally should. I went to Alaska last summer for a conference, and down in Seward during the summer, they have some of the dogs that are normally Iditarod racers, and they do their training in the summer down in Seward. So they have the National Historical Trail, and they really celebrate this whole idea of saving the children. So here's a little bit more of the story. The trail was originally used by Alaska Natives, used extensively during the gold rush, and then a portion of the trail was used by 20 mushers to relay diphtheria serum to Nome in 1925. So it commemorates that infectious disease aspect. It commemorates the original native history of Alaska, gold rush, etc. And of course, this facility has the adult dogs, and then they have puppies. They will not sell you the puppies. I tried to sneak a puppy. They will not let you sneak a puppy, but they're very, very cute. They will only sell the dogs once the dogs have developed a personality, because generally people who do sled, who use sled dogs, every position of the line of dogs has to have a different personality. The ones that pull, like closest to the sled, usually are dumb and strong. The ones in the front are kind of lazy but smart, so you have to figure out what personality is the dog going to have. And then you can do a fake little dog ride.
Let's go! Let's go!
And so even though I was there for a conference, I had saved up some money. I've never been to Alaska. So I saved up some money to do some of this fun stuff, like a little dog sled route. And so I thought it was really cool, and I thought I would share it with you, the idea that microbiology does play an impact in other parts of life as well.